The broadcast is now starting. All attendees are in listen-only mode.
Hello, everybody, and welcome to the webinar today. So I'm Ray from the Orange County R user group, and we're going to talk about apcluster. So we're here with Ulrich Bodenhofer from Johannes Kepler University in Linz, Austria. He is the package maintainer of apcluster. So happy to have Ulrich here. And I'm just going to go through some quick logistics stuff before we get into Ulrich's presentation. So if you look at my PowerPoint slide here, I'm going to talk about the GoToWebinar control panel. And we have an orange button on the control panel, and this will maximize and minimize the control panel. And once it's maximized, after clicking this orange button you see here with the arrow, you can do different things like select audio type. Most people are going to use microphone and speakers. If you're in the United States, you can use telephone mode as well. Try to close other applications you have open, things like LinkedIn, Skype, Twitter, Facebook, that can take bandwidth from the webinar. And then you can make your webinar full screen so you can see Ulrich's screen as large as possible. And then try to use headphones if you have them. Like iPod earbuds work well, or if you have a headset, that'll work well as well. So you notice that there's a questions pane when you maximize your GoToMeeting control panel by hitting the orange button, and there's a questions pane there. So you can ask questions if you have any questions, and we're going to do a question and answer session at the end of the webinar. So you can submit your questions here. A little bit about myself. So I'm Ray DiGiacomo Jr., president of the Orange County R user group in Southern California. And I'm a master art trainer and healthcare predictive analytics consultant. If you're interested in the Orange County R user group, you can join us on LinkedIn groups. So feel free to do that. And this webinar is currently being recorded. And you'll get an email today with a URL to the recording. And the recording should be up on the internet within the next 7 days, probably sooner than that, but give us at least 7 days to post it. So I'm going to introduce Ulrich Bodenhofer from Johannes Kepler University. He's a professor there, also the package maintainer of apcluster. And welcome, Ulrich.
Hi, everybody.
So what I'm going to do here is make Ulrich the presenter, and then you can go ahead and show your screen when you're ready. Great.
Okay, do you see it?
Yeah, I can see your screen. I see your slides up. And go ahead and introduce yourself and get started when you are ready.
Okay, thanks very much for the introduction and for the organization of this webinar and the invitation, Ray. Welcome everybody out there, wherever you are. I saw the prospective list of participants, and it seems that this is quite an international session today. People from all over the world seem to be interested in our package, which is very nice, which I appreciate very much. As Ray already said, I'm a professor in bioinformatics at Johannes Kepler University in Linz, Austria, right in the center of Europe. And I'm the maintainer of the apcluster package. And I also wrote quite a share of code in the package. As the name suggests, apcluster is mainly about affinity propagation, which is a clustering method. And today I will talk a little bit about the method and the package. Okay, so this is the outline which you probably saw in the invitation. So I will first talk a little bit about what affinity propagation is, and then I will talk about the package, and I will do a demo. And finally, there will be time for questions and answers. Okay, so what is affinity propagation? It's relatively new compared to classic techniques. It's a relatively new clustering technique, and I think it's justified to say that it has a strongly increasing importance in many fields, not only in bioinformatics but in many others. It has some very special features and some advantages over classical clustering methods, and that's probably the reason why it has increasing importance. One of the special features is that it's similarity-based, so that means you don't need a vector space structure for clustering data. You just need pairwise similarities, and this is something that you can have quite easily, even if you have structured data and really complex data. It's most of the time relatively easy to have some sort of similarity between items, whereas it may be quite hard to map complex data structures like chemical compounds or biological sequences or images or whatever to a vectorial structure. That's one of the special features, and the second special feature is that it's exemplar based. What does that mean? That for each cluster you get one sample that is really representative for the cluster, and it's not like a center in k-means, which is a kind of hypothetical average of samples in the cluster. No, it is one of the samples that you actually clustered. So it's a real sample. It's something that exists. Whereas if you map a biological sequence to a vectorial structure and then do an average, then you have an average that doesn't really correspond to a sequence that really exists. Affinity propagation is based on an iterative message passing scheme in which the samples compete for becoming examples. I will talk a little bit about that later on. One thing I have to emphasize here is that I'm not a developer of the method itself. It has been published. I will present you the reference in a minute. It has been published by Brenton Frey and Delbert Duc, both from the University of Toronto. And this paper got a lot of attention, and they provided MATLAB code for this method. And since I thought that it would be very helpful to have it available for bioinformatics applications, I thought it would be a good idea to have an R version of it, since R is quite common in bioinformatics, but MATLAB isn't. So that was actually the history or the motivation why we did a package. So we started from a simple port of the original MATLAB code of Frey and Duc, and then later on extended the package. And since well, for the last 3 years, the package has been growing. But you will see all the stuff in the package in a couple of minutes. Okay, so what's special about affinity propagation? As I already told you, it finds these exemplars, these representatives of clusters, to find the best, to find the optimal solution of exemplars is actually an NP hard problem. So it's something that you cannot do really meaningfully on a computer system, and affinity propagation finds a pretty nice approximate solution in really short time. So that's one of the specialties of affinity propagation. The second thing is that it's more or less almost a deterministic initialization independent algorithm that employs very simple update rules. And that it's initialization dependent and deterministic means that it has a great advantage over k-means, where actually the results of k-means pretty much depend on what you start from, and you may have to run k-means many times to get a decent solution. Not so for affinity propagation, which always gives the same solution in a deterministic fashion. And one side remark, what's also special about it is that the paper has been published in Science, which is quite remarkable for a data analysis method. So you seldom see papers in Science about methods like those. So it seems there was really sort of big interest and a big expected impact of this method that it got published in Science. Okay, just a few technical remarks. Actually, it's as I told you, a message passing scheme. There are two sort of messages. They're not actually exchanged. I would rather call it a metaphor, but I think it's a nice metaphor to say that that samples exchange messages. So internally, there's a responsibility matrix and an availability matrix, and the responsibility matrix corresponds to the accumulated evidence, how well suited one sample, or the kth sample, is to serve as the exemplar of I. Taking into account other possible exemplars. So it's the gathered evidence how well K can be an exemplar in a crowd of other samples, and if the availability matrix is something that is dual to it. So it's the evidence for how appropriate it is for I to choose K as its exemplar, but this time in just the other way around, taking into account other points that K would choose K as exemplar. So we actually have those two matrices for all I and all K, where I and K go from 1 to the number of samples, and these are the really very simple update rules. They're just 3 lines. In R, it's a little bit more than that, but not too much. Yeah? So it's just three simple rules that you have to implement. I'm not going into details here because it would take too much time, and it's probably not what you're primarily interested in. It's just one little detail here that you see this update for the responsibility matrix is done for all pairs of samples, even if I equals K. Whereas for the update of the availability matrix, we have a special rule for the diagonal and another rule for the off diagonal. And for the update of the responsibility, we have one particular thing here, namely if I is K, we have the diagonal of the similarity matrix. So S is the similarity matrix, and this self-similarities SKK are very important to control how many clusters you get. So these self-similarities are in the AP lingo called input preferences, and the higher they are, the higher the self-responsibility is, which means the higher these input preferences are, the more exemplars you get, so the more clusters you get. And the lower this value is, the less clusters you get. I will talk about this later on. It's just that you have an idea how these values influence the algorithm. So here is a little animation. You see a couple of samples. The strength of the color here in the left panel corresponds to how high the responsibility between pairs of samples is. This is correspondingly the availability matrix, and if you sum both together, then you get something like a unified view of which samples choose which exemplar. And you see that this is going over time, how after a couple of iterations, this nice cluster structure here emerges. Yeah? Okay, in our package, the most important function is apcluster. This is not the exact interface of it. I just picked out the most important parameters, because otherwise it would take too long and probably provide you with unnecessary details. So actually, what this function needs is, at first place, either a matrix of pairwise similarities, a quadratic matrix of pairwise similarities, or if you don't want to pre-compute the similarity matrix, you can provide here as the first argument a similarity measure, a function that measures similarity, that computes the similarity for pairs of samples. And then, of course, you have to provide data as the second argument. It can be a vector, it can be a list, it can be a matrix. It just needs to be some subsettable structure. Okay. And then we have two parameters. One is the input preference. That's exactly what I mentioned a minute ago. That's the parameter that adjusts the number of clusters. And alternatively, for finding a good setting of P, there is another parameter Q, but I will talk about that later on. This package is based on S4 classes, so the function returns an S4 object. The class is called apResult. It contains basically all of the stuff that is necessary to evaluate the clustering result later on. So where is light, there is shadows. So what are the pros and cons of affinity propagation? I think one of the great advantages is that exemplars are real samples and no hypothetical averages. I will show you some examples where it becomes, I hope, quite clear why this may be a great advantage. Another advantage is that it's only necessary to have pairwise similarities. And actually, there are no special conditions about what this similarity should satisfy. Can be more or less anything, need not even be symmetric or satisfy the triangle inequality or anything like that. So it's applicable to all sorts of kernels that you may know from different areas, correlation measures, anything. So more or less anything is allowed that gives a larger value if two samples are more related or more similar to each other, and gives a lower value if two samples are less similar. Apart from that, there are no other requirements. As I already said, the algorithm is almost deterministic. It's not sensitive to initialization. And one thing which you can consider an advantage, but it may also be a disadvantage, is that the number of clusters need not and cannot be pre-specified. That means under many conditions, you get a nice number of clusters, but if you are interested in a particular number of clusters, it may be tricky to find a good setting for that. But I will talk about that later on. That's exactly the first minus here, that the adjustment of the input preference parameter, which influences the number of clusters, can be tricky. One more important point from a practical standpoint is that in order to run affinity propagation, you need a quadratic similarity matrix. So if you have 1,000 samples, you have 1,000 by 1,000 similarity matrix. And if you have 10,000 samples, you would need a 10,000 by 10,000 similarity matrix, which poses a serious limitation in terms of the size of the data set, which is not a pleasant thing. So in these times where everybody's talking about big data, this is probably not a very good thing. But I will tell you also one possibility to overcome that limitation. Okay, about the adjustment of input preferences or the number of clusters. So, well, you can always, as we have it here, you can always adjust P, yeah? But actually P is, it's quite hard to determine a good P. It's a sort of dimensionless, it's on a dimensionless scale, and it's very hard to find a good value. You just by thinking about it, it's too hard. There is the rule of thumb that has been published along with the apcluster paper by Fry and Duck, and they said to have some moderate number of clusters, choose P as the median of off-diagonal similarities. Don't specify P when you call apcluster, it automatically determines the median of off-diagonal similarities and makes the setting. But apart from that, you can directly influence depending on the distribution of the similarities, you can directly influence what is chosen with the Q parameter. So Q parameter is that the Q parameter does the following. It sets the input preference to a quantile of the off-diagonal similarities. So if Q is 0.5, then this corresponds to the median of similarities. If Q is 0, then it corresponds to the minimum of similarities. So we have all these rules that the original authors of the affinity propagation method suggested in there, and smooth transition between them. Okay, that's why I think that's actually a new parameter that we introduced, and I think it's a very nice thing, because without knowing anything about which values are in this similarity matrix, without knowing which would be good choices for P, you can really very easily adjust in a range from 0 to 1 the number of clusters. Apart from that, there's a function preference range that computes bounds for the input preference. So in case that no setting of Q gives a decent result, you may run preference range on your similarity matrix, and this gives you two values: a lower bound for P, at which you will get 1 or 2 clusters, and an upper bound for which you will get as many clusters as you have samples. Okay. Apart from that, there is a function apcluster k. It basically works exactly like apcluster, but you can specify how many clusters you want. And what happens is that an inverse search is performed to find an input preference value that yields exactly this desired number of clusters. Yeah? And there is one more thing, which is actually my development. It's something I developed. It's a function called apcluster. It's an agglomerative clustering that is exemplar based. So it follows a similar philosophy as apcluster, where each cluster has one dedicated exemplar that is hopefully representative for the cluster. So it's an agglomerative, it's a hierarchical clustering method, and you can not only use it on the similarity matrix or your raw data directly, you can also use it on top of an AP clustering. So if you have a result of affinity propagation that has I don't know, slightly too many clusters, you can perform an agglomerative clustering to group some of them into larger clusters, and then you can actually specify certain cut levels with a number of desired clusters as well. I will show you a little bit about this later on. Okay, so the question remains: what do we do with large data sets? So one trick could be subsampling. That means we select some samples and perform clustering on this subsample. So if we have this quadratic similarity matrix here, and we select, let's say, the samples that correspond to the gray stripes here, then we will pick out those samples both on this axis and on this axis. And actually, the similarity values we take into account finally are the dark gray ones here. And actually, if you do subsampling, you lose a lot of information, and you probably lose a lot of information about the cluster structure. And the idea of a method that was also developed by Frey and Duck is to consider all objects for clustering, but just to limit the number of possible exemplars to a subsample. So what we do actually is, if we go back here, we don't reduce row and column objects when we do subsampling. We actually only reduce column objects. That means we limit those samples that may be chosen as an exemplar, but we still cluster all data and we still take all data into account. And That's the idea of what is called leveraged affinity propagation. And it starts with a small but hopefully reasonable subsample of columns, so potential exemplars, and iteratively repeats this. Actually, affinity propagation, it's just a slightly generalized affinity propagation that is done on these non-quadratic similarity matrices. And this subsampling is repeated iteratively, but it's not redone every time. In each iteration, you preserve the examples of the previous iteration. That means you acquire additional evidence for what is a good exemplar without losing what you have considered previously. Yeah? And that's why you don't need to calculate the whole similarity matrix in advance. So that means, for instance, if you take 1% of your data as potential exemplars, then you have just 1% of the similarity matrix. And if you do it, even if you do it 100 times, which is maybe a little bit exaggerated, but then you actually have 100 times 1% of the similarity matrix, and you don't have to keep the whole similarity matrix in your memory. That it's just 1% of memory that you need. We have this function apcluster. It basically works exactly like the apcluster function. So it starts from a similarity measure and the data. Of course, it cannot start from a pre-computed similarity matrix of a complete dense quadratic similarity matrix, because if you have that, it doesn't make sense to use leveraged affinity propagation. You can use regular affinity propagation. We have the same parameters P and Q, and apart from that, we have frac and sweeps. Frac is the relative proportion of samples that you take in each iteration. So if you have if you want 1% of column objects in each iteration to be considered as potential exemplars, then set frac to 0.01. And sweeps is just the number of times you repeat this. Okay, we will see all this when in the demo session later on. Okay, we have plotting functions. We can, this is a detail, I skipped that. You can plot the dendrogram of an agglomerative clustering. You can plot an AP result object along with the original data. And of course, you can plot heat maps of clustering results. We have 4 built-in similarity measures, negative distances or negative quadratic distances. That is one of the classic things, also suggested by Frey and Duck. Then exponential similarity matrix, that's like a Gauss or Laplace similarity. Then we have these truncated linear similarities and correlation measures and so on. So this is just to visualize quickly. This is what the negative squared Euclidean distance looks like, which is a standard setting, or that's a Manhattan distance. It takes a while. This is Gauss and a sort of Laplace distance. What comes next are these truncated inverted distances. It's just something like 1 minus a distance truncated at 0. It's just okay. It's if you take 1 minus the Euclidean distance and truncate it at 0, you get such a cone. Or if you do this for the maximum distance, you get this pyramid. It's just a little bit of play. By the way, these graphics were not created in R, but in Mathematica. I'm sorry for that. In terms of 3D plots, I think Mathematica still produces prettier outputs, and I hope that R will one day catch up with that. So here is an example that I did myself, actually. You see here two clusters determined by affinity propagation of coiled coil sequences. I'm not going into much detail what a coiled coil is. It's just a special type of protein where you have these double helices like shown here. And the interesting thing is that the exemplars are shown in boldface. And if you look at the patterns that are most indicative for this type of sequence and this type of sequence, this is really a very good exemplar, because it has all the patterns inside that are indicative for this cluster. What we also did here, a colleague of mine was that's only a toy example, but that's a clustering of chemical compounds. You see all the names here, and I think the heat map pretty much shows that this is a quite good clustering. So this is really complex data. We have a graph structure actually on which we have defined a graph similarity measure. I think it was some Tanimoto distance or something like that for people who are working in this field. And you can directly use apcluster with that and get nice results. What happens if you do leveraged affinity propagation, then you see that in this case you have much less potential samples that are taken into account as exemplars, whereas on the other side you have all the samples. This is just a little toy example. Okay, it's time for the demo, right?
Yes, thank you for that great lecture. That's very interesting stuff.
Thank you. Okay, so the package is actually on CRAN, which means everybody can install the package just by typing in install packages apcluster, or depending on what sort of R version you have, there may be even some menu items that you can use. Okay, so first we have to load the package. You see it's based on RCPP, which is a really cool package for integrating C++ code into R. Okay, now I'm creating a simple 2D data set consisting of 4 Gauss clusters. So, sorry. Okay, so it's something like that. So we have 1, 2, 3, 4 clusters. Now let's run apcluster. So it just takes an instant. So see here, in this case, 5 clusters have been identified. This is how the AP result object is being shown. So you have the number of samples, how long it took, the input preference that has been used. This is with the default setting that I told you about, that P is chosen as the median of the similarities. We have the number of clusters and these things, which samples have been chosen as exemplars, and then even the clusters are shown. So for each exemplar, you see the number, you see all the indices of samples. Okay, so now let's show the result. So here you see these are the 5 clusters that have been identified. And this is the heat map corresponding to it. So actually you see, well, maybe the input preference is slightly too high. It looks pretty much like there's 4 clusters. So let's try Q equal to 0, which is the minimum actually, and now we have 4 clusters, and I think that's pretty much what we would expect. And now in the heat map you see exactly these 4 clusters, and to the left and on top of the heat map you even see the dendrogram. If you merge these clusters further on with this AggX cluster method that I told you about, then this heat map, this dendrogram would be obtained. Okay, then now let's try another data set with less separated clusters, with quite ambiguous clusters. So the question is, are these 2 clusters, are these 3 clusters, are these 4 clusters? Well, they were created as 4 clusters, but typically clustering methods wouldn't be that sure about it. Well, but apcluster in this case actually found 4 clusters. So let's plot them. So that's a very cool result, and that's the heat map. And here in the heat map you see that 2 of those clusters are actually pretty close to each other, and 2 others are also pretty close to each other, but there seem to be 2 groups of clusters, so 1 and 2 and 3 and 4, and which are quite clearly separated from each other. If we plot the data set again, I think this is exactly what we're seeing here. Okay, now let's create a 4D data set. I'm doing this just by joining the previous two data sets. Again, apcluster takes more or less no time. We again get 4 clusters. Okay, but how can we plot a 4-dimensional data set? Well, what we can always do is to plot the cluster structure on 2 columns of the data. That's one thing that we would have here. These are the columns 1 and 4, so the first and the 4th dimension. But what the package also can do is to create a scatter plot matrix and superimpose the clustering result on the scatter plot matrix. That's what you see here. This is actually a very new feature that we included in version 1.3.2, which has been released just yesterday. So if you install the package now, then you can already get the new version. Okay. Well, the heat map is independent of the dimension anyway, so we see very nice clusters here. Okay. What happens if I make the whole thing a little bit more complicated? So let's do something similar to previously, but now we add 1 column that is pure noise. Okay, apcluster really works fast. And now you see that suddenly, because of this noise column, we get many more clusters. So, and the structure looks pretty messy, actually. So, because of the noise column that stretches clusters apart, actually, apcluster identifies many more clusters. And I think also the heat map is not that clear anymore about how many clusters there are. It looks as if there were 8 or 9 clusters here. Okay, let's try this again with the setting q equal to 0, and then we get 5 clusters. Yeah, and still in some of the dimensions, this clustering result actually looks quite nice. Let me try this preference range parameter. As I told you, it gives you lower and upper bounds for the parameter p. And here you see that minus 14.5 is the lower bound, and almost 0 is the upper bound. And now let's see what happens if we, for instance, choose p equal to -2, and then we get 4 clusters actually. What if we want to enforce a particular number of clusters? I already told you for this we have the apcluster k function. It works exactly like the other functions, apart from the parameter k. And then some inverse search is taking place, which tries to find a good value for p that gives exactly this number of clusters. Yeah, and I told the method to find a clustering with 5 clusters, and that's actually what we get. Okay, I also told you about apcluster, which is an agglomerative method that can be used to further cluster 2 fine-grained clusterings made with affinity propagation. I ran this on the one with the 8 clusters, if I remember correctly. Yes, that was the one with the 8 clusters. And then these 8 clusters are further merged. And I think from this dendrogram, it really looks as if there were 2 groups of clusters. Actually, it pretty much looks like 4 clusters that are grouped into 2 larger subclusters. And what I can do is, for instance, I can cut this dendrogram at this level here, to get 4 clusters, and this is what comes out. And I think this looks pretty good. Yeah. Okay. Finally, let's try something less trivial. These were all toy examples. Maybe you're interested in a more advanced. I'm really sorry for that. I should have checked that before, but it will take only a second.
Yeah, we got some time, so we don't have to rush.
I'm really sorry for that. It's the computer at home, and okay, there it is. Okay, sorry, it was just a short interruption. Okay, there is a test data set containing biological sequences that's included in the apcluster package. So we have the promoter sequences of a few human genes, and what you see here, for instance, they are named by the I think the RefSeq identifiers of these genes. So these are DNA sequences. And if we want to cluster such sequences, it's no problem with apcluster. So for instance, I can use the spectrum kernel, which is a kernel that is based on common substrings, in this case of length 6. Okay, so here we have the similarity matrix of the first five samples here, and well, okay, you see these are, I think, 150 samples, and it just takes an instant. And finally, 8 clusters have been identified. So that's actually the heat map. The sequences are quite dissimilar, actually. And this is not to make a biological point, actually. This is just to show you how easily this can be done. So it's questionable whether there are actually 8 clusters or more or less. I don't know, but it's just to give you an idea how easy it is to apply affinity propagation. So just assume for a moment that we are not satisfied with these 8 clusters. So let's say we want to join some of them. So that would come out if we further group them together by this hierarchical method AggXcluster. Then you see, well, maybe there are 5 clusters if we adhere to that viewpoint for a moment. Okay, I can do this. I can just cut the tree at 5, and then we get 5 clusters, and this is the resulting heat map with the 5 clusters. So you can really play around and explore, and that's really pretty easy to do. Now let's ask the question how long it takes if we have a larger data set. This is not particularly large, but at least much larger than the ones we had. So here we have a data set with 4 clusters, but this time we have, as you see, we have 1,750 samples. If we run affinity propagation, it takes, so this is regular affinity propagation with the quadratic similarity matrix. This runs for at least a few seconds. And then we will try leveraged affinity propagation, and you will hopefully see that it takes much less time. Probably only a few more seconds to complete. Yeah, there we are. Yeah, interestingly, we have very many clusters. That's something that happens quite often when you do affinity propagation, that if you have a very dense data set, then you get maybe a little bit too many subclusters. So that also takes a while because there are pretty many items to display. Yeah. Now let's try preference range. We see that these are the values in which we may choose the affinity propagate the input preference. I'm not running this again because it will take much time. I will run leveraged affinity propagation instead with 5% column objects, and I run this 3 times, and you see that it just took a few seconds, and I got 4 clusters, which is exactly what I wanted, and this is the result. And once again, already finished. So this is really a great speed up compared with the standard affinity propagation that takes all possible combinations into account. So here we had 5%, and now let's try 1%, which takes even less time, gives the same number of 4 clusters, and the result is more or less the same. It's probably a little bit questionable what happens with these here, but maybe if I run this more times, maybe we can get rid of this artifact. Let's see. No, now it's gone. If I do more sweeps, then the chance is better to find a really good solution. What is the limiting factor in terms of how much memory you need is actually frac. And if you use, as in this case, only 1% of the data, you have a very limited view of the data. But if you do it sufficiently many times, then you also get a decent solution. Okay, so that's more or less all I wanted to show you here. Let's move back to my slides. So there is just a few more things that I want to tell you. We have a paper in Bioinformatics about the package, and if you use the package, I would really appreciate it if you cited the paper. If you need more information about the package, this is the homepage, and this is the page at CRAN. And finally, I want to acknowledge my package co-authors, 2 students, Andreas Kotmeier and Johannes Palme. Some more people at my institute who supported me with examples and some other types of help. And finally, I want to thank Ray and the Orange County R user group for the opportunity to speak to you here. Thank you very much.
Yeah, you're very welcome, Ulrich, and that was a very exciting webinar, so thank you for your time, and we're glad to have you.
You're very welcome.
Yeah, so well, let's do some question and answer now. We have about 10 minutes left in the hour.
Yeah.
So I have a question here from Alvaro, and Alvaro was using apcluster and ran into some memory limitations. I'm not actually sure what version Alvaro was using of apcluster, but Alvaro mentioned the data set was maybe 200,000 data points, and or excuse me, yeah, 200,000 data points, 6 features per vector, and he was on an iMac with about 16 gigs of RAM, and had some memory limitations, and wanted to know, are there specific size limitations that apcluster has, and are there different versions of apcluster that can take advantage of, say, multi-cores or multi-clustered systems? So mainly just memory limitations. Yeah.
Yep. Very good question. So first of all, you see that if you use regular affinity propagation, then you have really a squared matrix of similarities. And you see here, if you have 200,000 samples, then you would have 4 times 10 to the power of 10 memory cells that you need, where each of these cells is 8 bytes. So that's actually pretty much, and prior to version R30, I suppose it was not even possible to create a data object of that size. It may be possible in R30, but still I wouldn't advise you to do that. So if you have that many samples, it will also take way too much time to cluster the samples. So this is exactly the type of problem for which leveraged affinity propagation has been designed. And I think the leveraged affinity propagation has been integrated into the apcluster package with version 1.3.0, which was released in January. So maybe Alvaro hasn't seen it yet. I don't know. So please try leveraged affinity propagation, which is in one of the newer versions of the package. And if you still encounter some problems, please get in contact with me. Well, that's also a message to all the others. If you have any problems or questions related to the package, you can contact me anytime. And so far, I have always answered all these emails, and I have made contact with a lot of people, which I appreciate very much, that they use my package and that they also dare to ask me questions, and I'm really glad to answer them.
Thank you. So we have another question from Paul, and Paul noticed that you were doing DNA clustering during your presentation, and wanted to know if this same theory can be used in corporations, for example, transcripts in corporate call centers. You know, when you call in for customer service and they type your question into a database, can you take those words and do clustering of those words?
Yeah, sure. I mean, it's a matter of how you represent this data. Yeah? You can use a vectorial representation with word counts, maybe after stemming, or you can just take the texts and compute some similarity of the texts, maybe based on single words, or maybe based on the co-occurrence of words. I'm not an expert in text processing to know what are good similarity measures, but well, yes, of course. As soon as you have a similarity measure for your data items, you can use apcluster anytime. It's promiscuous in the sense that it can handle all sorts of data if you have a similarity measure. And that's one important aspect. Thanks for this question that I haven't mentioned. apcluster is of course not limited to the similarity measures that I showed you that are integrated in the package. So you can write your own similarity measures and use them. Either you compute the similarity matrix first and run apcluster on the similarity matrix, or you can also do the following. You can also write a function that computes the similarity. You have to do this according to a special specification, which is documented in the vignette of the package. And you can even run a leveraged affinity propagation using your similarity function. So, and it's not limited to text or DNA or whatever. You can use it for chemical structures, what we have done previously, or images, signals, whatever. It's completely up to you.
Great, thank you. So could you talk about the apcluster's ability to handle missing data?
Yes. So we just made a slight change to that in the latest version that we released yesterday. Before that, the package could already handle missing data, so NAs in the similarity matrix, but in the latest version it has been slightly improved. The idea is that if there is an NA, a missing value in your similarity matrix, then this pair of samples is simply not considered. So a sample cannot choose another sample as exemplar if the similarity is unknown. So you don't know how similar these samples are. Therefore, you shouldn't make any point about one could represent the other. And as long as there are samples that stand or that can be representatives, exemplars for many others, it shouldn't be a big deal. If there's a large percentage of missing values, then affinity propagation cannot handle that. You would probably get a lot of islands and a lot of clusters, or also maybe strange phenomena that remote samples will be clustered together just because only the small similarities are known, but all the other similarities are unknown. So strange things can happen if you have large numbers of missing values, but if you have a moderate number of missing values, something like up to 5% or so, it shouldn't be a problem, and you should still get a decent result.
Great. So we have about 2 minutes left in the hour. If you have a question for Ulrich, please submit it now. So this is the last chance to submit your questions through the chat window. So just a couple more questions here. Could you talk about apcluster's ability to handle sparse matrices?
Well, currently we do not support sparse matrices. It has been on our list for quite a while.
Okay.
So if you have a sparse similarity matrix, you can always convert it to a dense similarity matrix with making some hopefully meaningful setting where the sparse matrix didn't have any entries, so maybe minus infinity or NA or something like that. So, but presently, no, we don't have that, but it's on our list.
Okay. And can a person run apcluster with a custom similarity measure?
Yeah, that's what I already mentioned. That so you can use the built-in ones. You can use whatever method you like to create a similarity matrix and run apcluster on similarity matrix, but you can also write a function that computes similarities and run apcluster or leveraged affinity propagation using the similarity measure.
Okay. Well, great. Well, we're at the end of the hour now, so thank you, Ulrich, for your time. It was a great webinar.
You're very, very welcome.
Yeah. So is there anything you wanted to say, Ulrich, to the audience before we close down the webinar?
Well, there's not too much to say, just that I thank everybody for the interest and
Sure.
for attending the webinar, and I would appreciate if you use the package, and if you have any question or problem, don't hesitate to contact me.
Well, thank you. Everybody in the audience will be getting an automatic email from the GoToWebinar software. And this will have a link to a location where the video will be posted. So if you had any friends that could not attend, the video is being recorded now. It should be posted within the next 7 days, probably sooner than that, but give us about 7 days. And I will go ahead and close down the webinar now. So thank you everybody for coming. Have a good afternoon, morning, or evening, wherever you are in the world. And we're going to go ahead and shut it down. So take care, everybody, and thank you for coming.
Thank you.